Hello. With this video, let's try to understand endomembrane system. Endo here refers to something that is inside, and membrane here refers to a covering which is surrounding an internal structure. So endomembrane here refers to membrane-bound structure. System usually refer to an entity which is constituted by many parts, and all these different parts are working together. In case of our digestive system, we have alimentary canal along with other organs which are working together in systematic manner. So system here refers to composed constituted by many parts, and all these parts are working together collectively. So endomembrane system is usually referring to cell organelles, some cell organelles which are usually membrane bound, and these are working together in coordinated manner. Coordinated manner here refers to which are walking together collectively, and their walks are usually synchronized, or they function together so that they match their functions, and finally they usually perform a collective function, a major function, which is usually collective product of all the cell organelles which are having membrane bound. So endomembrane system includes endoplasmic reticulum, Golgi apparatus, lysosome, and vacuole. All these are membrane-bound cell organelles, and they are working together cooperatively, and their work is usually coordinated. And this topic, that endomembrane system, is not mentioned in your textbook, but it's important. I found it important to mention here so that later you may not face problem, and it's easier for us to understand also. So endomembrane system is a topic which is discussed under chapter 5, the fundamental unit of life. So let's see the different endomembrane structures which are present within the cell. So this is a diagrammatic representation. The diagram is drawn with the help of PPT on computer by me. So we have endoplasmic reticulum, Golgi apparatus, lysosome, and vacuole. All these are endomembrane system. This is endoplasmic reticulum. Endoplasmic reticulum is rough or smooth endoplasmic reticulum, and these are usually near around the nucleus. We also have a Golgi apparatus near to the nucleus. We have lysosomes. We have we have vacuole. So all these are working together in collective manner, or in coordinated, synchronized, or in cooperative manner. Suppose usually endoplasmic reticulum synthesize protein and lipids. So suppose this image is coming, okay. So lipids and proteins are usually synthesized by endoplasmic reticulum, which may be rough or smooth. Later on, it will be transported to Golgi apparatus. After processing it, it may be forming vacuole or lysosome, or these may be transported to your different cellular structures for building up their structure. So for instance, I'm showing you 3 dots here. These are representing proteins or lipids synthesized by endoplasmic reticulums. So if you usually these endoplasmic reticulum, Golgi apparatus, lysosome, and vacuole, all these are working together. So in order to synthesize cell membrane, first it requires protein and lipids. Cell membrane is lipid bilayer. It requires protein and lipid, mainly lipids. So lipids and proteins which are synthesized by endoplasmic reticulum will be processed by your Golgi apparatus and send it to your cell membrane for making or building up cell membrane. So this is how your membrane systems work together. Now let's see one by one. First we have endoplasmic reticulum. Endo refers to inside, plasmic refers to plasma or fluid. Reticulum here refers to network of structures. So endoplasmic reticulum is having network of structures, and these network of structures are usually tubular in structure. Let's see, suppose this is a nucleus. Around the nucleus we find endoplasmic reticulum. Endoplasmic reticulum may be having ribosomes on it, here represented by black dots. So ribosome having endoplasmic reticulum are usually called as rough endoplasmic reticulum because it's appearing very rough under microscope. We also have smooth endoplasmic reticulum, which is without having ribosome, and these are called smooth endoplasmic reticulum. Function of rough endoplasmic reticulum is to synthesize protein. Usually proteins may be enzymes, hormones. So enzymes and hormones are secreted out by rough endoplasmic reticulum. Smooth endoplasmic reticulum is responsible. Smooth endoplasmic reticulum without ribosome and appears smooth under microscope. That's why smooth endoplasmic reticulum. And it is producing fats or lipids. So usually endoplasmic reticulum is the site for synthesis of fat molecule. It manufactures, it is producing lipids or fat molecules. So this is what we have discussed. We had seen endoplasmic reticulum having membrane, and these are usually tiny tubular structures or sheet-like structures, usually scattered in the cytoplasm around the nucleus. So you can see sheet-like structure or tubular structures which are connected together forming a network. So endoplasmic reticulum is having membrane, and these are tiny tube-like structures, or these appears like sheets in the cytoplasm around the nucleus. We have rough endoplasmic reticulum having ribosomes on its surface. Smooth endoplasmic reticulum usually reticulum is not having ribosomes on it. Now we had already understood, if we have understood the structure, then we are indirectly able understanding the functions. We also have discussed the functions. So rough endoplasmic reticulum is actively involved in protein synthesis. Proteins may be enzymes or hormones, so these are synthesized by rough endoplasmic reticulum and secreted, produced. Smooth endoplasmic reticulum is mainly responsible for production of fat molecules or lipids. There is another essential role of smooth endoplasmic reticulum in the liver cells. Liver cells are having smooth endoplasmic reticulum which are capable of detoxifying poisons and drugs. Detoxification here, or detoxifying mechanism here, refers to removal of toxic substances. Toxic substances are harmful. These are usually these should be removed out from our system. So liver cells are helping in D here refers to removal, helping in removal of toxic substances. So with this we have understood the function structure of endoplasmic reticulum. Now let's see. Yes, we can also say that, collectively say that endoplasmic reticulum is helping in production of protein and lipids, and these are usually processed and finally transported to Golgi apparatus, or these are forming lysosomes or vacuole. So these are also helping in formation of cell membrane, membrane biogenesis we had already discussed earlier. So endoplasmic reticulum is helping in production, it is helping in processing, here refers to refining and transport of proteins and lipids, so that these will be forming Golgi apparatus or cell membrane or vacuole or lysosome. Now let's try to find out what endoplasmic Golgi apparatus looks like. First, let's try to pay homage to Camillo Golgi. He's the scientist who had discovered Golgi apparatus for the first time. He's a great neuroscientist. He's researching on nervous system, cells of the nervous system, that is neuron or nerve cell. While he was researching about neuron, he found a densely stained here refers to colored. So densely here refers to darkly stained, densely colored network of structures within the nucleus, and he called it apparatus. Darkly colored structures in network, and he called it apparatus. Later, after his name, now we are calling Golgi apparatus after him, the scientist who had discovered Golgi apparatus for the first time. Now let's see the structures of Golgi apparatus. Golgi apparatus is sometimes called as Golgi body or Golgi complex. Now this is how it looks like. This is a 3D diagrammatic representation. You can see flat disc-like structures, and these flat disc-like structures are called cisternae. One flat disc-like structure is called cisterna. This is a cross-section, cut section of your Golgi apparatus, and usually these are having structure like this, shape like this. These are having disc-like structure. So discs, you have seen CD disc is a disc, so oval shaped, having lumen inside, cavity inside, and it is a membrane bound. So flat disc-like structure. Over here it's shown in cross-section manner, cut manner. So you are also able to see secretory vesicles. Vesicle here refers to sac-like structure, which is having membrane, and these are tiny in nature. Golgi apparatus is having two surface or two faces. We have cis or forming face, where there is formation of secretory vesicles. We also have trans or maturing phase. Trans here refers to trans or maturing phase here refers to maturation of vesicles. So later on, from trans phase of Golgi apparatus, there is transportation of protein, processed or packaged proteins or lipids to other parts of the cell, or to lysosome, and for formation of lysosome and vacuoles. Now this diagram is little difficult for you to draw, and it's time consuming if you draw it. So to make it easier, I've come up with this diagram. This is also representing your Golgi apparatus. We have cisterna. We are also having secretory vesicles. We are having, okay, I've mentioned two secretory vesicles. Okay, it's fine. So vesicle here refers to sac-like structure having membrane around it. We also have two faces, two surfaces, cis or forming surface, trans or maturing surface. Now with this, let's try to summarize what we had studied about Golgi apparatus. Golgi body usually stores, modify and pack the products in vesicles. Golgi apparatus is helping in storing of lipids and proteins, and it is also modifying, changing the structure. It is also processing the protein and lipid, and finally it pack the lipid and proteins in the form of vesicles, in the form of membrane bound tiny structures. Some sugars like glycoproteins, glyco here refers to sugar, lipid here refers to fat molecules. Protein, we all know protein is combination, or it is polymer of amino acids. So glyco refers to sugar or carbohydrate. Usually Golgi apparatus is also helping in formation of glycoprotein and glycolipids. Now after budding of protein and lipid, there is formation of your lysosome, mainly proteins which are secreted out, which are stored, modified and packed by Golgi apparatus, is usually forming your lysosome. Lysosome is having enzymes, so enzymes are usually proteins. Now let's discuss about the suicide bags of cell. Lysosome is suicide bag of cell. Lysosome is having membrane bound structure. It is having it is usually a vesicle. Vesicles are usually having membrane so around it. So it is a membrane bound structures which are usually produced by Golgi apparatus, packed, formed by Golgi apparatus. And inside the membrane bound structures, you are, as the structure of membrane bound structure, you are able to see hydrolytic enzymes represented in dots. So these hydrolytic enzymes are also called as hydrolases, which may be lipases, fat digesting enzyme, protein digesting enzyme proteases, carbohydrate digesting enzymes, carbohydrases, and these are helping in these enzymes are helping in digestion of lipids, proteins, and carbohydrate at optimum pH. Okay, we have already discussed the last point. Hydrolases are capable of digesting, or these are helping in breaking down of carbohydrates. Carbohydrate is digested by carbohydrases, protein digested by proteases, lipids digested by lipases, and nucleic acids are digested by nucleases. So anyhow, lysosome is having membrane bound structure. These are tiny vesicles, sac-like structures within which we have hydrolytic enzymes. Now this is a big question. Why do we consider lysosomes as suicide bags of cell? Mainly because of the enzymes present within it. Once lysosome bursts, then there is releasing of all the enzymes, protein, carbohydrates, lipases, and even nucleases. All these enzymes, if come out of the lysosome after the bursting of lysosome, then these enzymes are going to digest the parts of the cells which are made up of carbohydrate, proteins, and lipids. You also have other cellular content. If these are digested, then cell will be dying. So we can say that lysosomes are known as suicide bags of the cell, mainly because of on bursting lysosome, hydrolytic enzymes, hydrolases like carbohydrates, proteases, lipases will be coming out into the cell, will be spilling into the cell cytoplasm. Later on, it will be destroying all the internal cellular cell organelles, and finally it will be also digesting the cell membrane. In that way, lysosome is not helping, is destroying the entire cell. So that's why we are considering lysosomes as suicide bags of the cell. Now, function of the lysosome, we had already discussed, but still then we try to make our learning more concrete, more constructive. So function of lysosome is usually involved in breaking down of waste or excess substances. There may be some cell organelles which are aged, or these are worn out. These are damaged cell organelles. If we have damaged cell organelles, these are usually digested or broken down into pieces by lysosome. We also have food particles which are also which are digested with the help of lysosome. Engulfed pathogens are also digested by lysosome. So lysosome is having enzymes. So enzymes will be helping in digestion of all this unwanted or waste substances or harmful substances, which harmful entities which are coming inside the cell. As it is breaking down waste, it is also indirectly referring to removing of waste. So lysosome is helping in removal of waste. So these are structures, these are functions of lysosome. Now discuss about the vacuole as endomembrane system. You can see vacuole is having simple structure, very simple structure. The membrane of the vacuole is known as tonoplast. Inside the membrane we have, in case of plant vacuole, we are having plant cell vacuole, we are having sap. Sap is combination of water. It is having organic ingredients. It is also having, say, like different nutrients. It may be having hormones. So sap is combination of water plus dissolved substances and plants. So vacuole is having membrane called tonoplast, and inside it we have fluid substances. In case of plant cell, we have vacuole which is having sap. So tonoplast is the membrane of vacuole, and in case of the plant cell, vacuole occupies 50 to 90% of the entire size of the cell. Usually vacuoles contain water, saps, amino acids, sugars, organic acids. Function of the vacuole is diverse. In case of amoeba, they are having contractile vacuole, which is important for excretion of excess substances or undigested food particles. So contractile vacuole is found in usually amoeba and other unicellular protists. Protists also do have foot vacuole, which are usually vacuole having foot inside it. Amoeba is engulfing food particles and there is formation of foot vacuole. So foot vacuole is usually having nutrients and which will be essential for the amoeba to survive. In case of plant vacuole, it is providing turgidity and rigidity to the cell. As plant vacuole is occupying about, say, like 90% of the entire cell size. If vacuoles are filled with water, vacuoles are filled with nutrients, sugars, so there is bulging of your vacuole, and thereby there is giving shape to the cell. So your vacuole is helping in giving turgidity and rigidity to the plant cells. Vacuoles also stores amino acids, sugar, various organic acids and proteins, so which are important for living, important for survival of the plant cells. So vacuoles are having nutrients also. So these are the structures of your these are the functions of your vacuole. Now let's try to answer these questions. You can answer these questions by your own. With this, you'll be also writing down in your fake copy. These answers should be written down in your fake copy. First question is, what are endomembrane system? Though not given in your textbook, but it's important for you to understand. You'll be able to understand easily, and it's good for your understanding. Second, analyze the endomembrane system. Third, you have to draw a well-labeled diagram of endoplasmic reticulum. Fourth, state the functions of rough endoplasmic reticulum and smooth endoplasmic reticulum. You also have to draw a diagram of Golgi apparatus. Without drawing diagram, we won't be able to learn the cellular structures. So sixth, you have to mention the functions of the Golgi apparatus. Functions are understood once we know how to draw, and after understanding the structures of the cell organelle, we can easily state the functions. Seventh, name the name the soot side bags, not name the call. Just name the suicide bags of cell. I'm saving it. So the seventh question is, you have to name the suicide bags of cell. Why do we call so for that organelle? So you have to mention why we are calling one particular structure which you are naming it is called as suicide bags of cell. You also have to discuss about the importance of the vacuole in cell. Ninth question, why do we collectively use term endomembrane system for cell organelles like endoplasmic reticulum, Golgi apparatus, lysosome and vacuole? 10th, you have to mention the importance of endomembrane system. So with this, I'm going to conclude some information about endomembrane system. You'll be learning more once you are promoted to higher classes. So that's all for today. Thank you. Stay safe. Take care of your health. Bye.